Vande Gujarat.
Hello friends, I am Dr. Archana Gattupalli. I am from MS University of Baroda Microbiology Department, and today I will be covering key concepts and problem solving in enzymology. This topic I am going to cover in two parts. Part 1 will cover general features of enzymes, enzyme classification and nomenclature, and enzyme assays. Part 2 will have Michaelis-Menten equation, enzyme inhibition, and certain other miscellaneous aspects about enzymology. In both these topics, I will give some important aspects or concepts which have to be remembered and understood by the student, and then followed by some problems that are related to that particular topic and the solution to that problem. So some of the important features of enzymes. We all know that enzymes are biological catalysts. By and large, they form the major catalysts of living systems. RNAs also catalyze reactions, but they are in small number. Majority of the catalysts, biological catalysts, are enzymes or proteins. They were discovered in 1900s by Buchner, and the name enzyme literally means in yeast. In human beings or in animal systems, approximately around 40,000 different enzymes catalyzing different reactions are present, whereas in small prokaryotic organisms like in E. coli also there are about 3,000 different enzymes that are present. The main important feature about enzymes is that they increase the rate of the reaction. And the rate of the reaction can be increased anywhere between a factor of around 1,000,000 times faster to 10 raised to 17 times faster than catalyzed reaction. So coming to the type of reactions, it is very important to understand that reactions can be spontaneous or they can be kinetically unfavorable reactions. The feature about whether a reaction is spontaneous or not is based upon its delta G, that is the free energy difference between the reactants and the products. A negative delta G makes the reaction thermodynamically or energetically favorable, whereas a positive delta G makes it kinetically unfavorable. So the spontaneous reactions, which are otherwise energetically favorable, need not always proceed at a fast rate. Reactions could be spontaneous and fast, such as the example given here, or reactions can be spontaneous but extremely slow. To speed up the reactions, the barrier of the activation energy has to be overcome. The transition state theory explains that why certain reactions are slow or certain reactions are fast, although they are accompanied by a negative delta G. So the barrier to be crossed is the energy barrier known as the activation energy, and at the peak of the activation energy is a state of the molecule which is termed as the transition state, which is the high energy state through which the reactants must pass in order to get converted to the products. Catalysts lower the activation energy. So we see here in this diagram that the activation energy barrier in the catalyzed reaction is lower as compared to the uncatalyzed reaction. So this difference between the catalyzed and the uncatalyzed reaction in the free energy of activation is known as the delta G star catalysis, and this implies that basically catalysts do not change the delta G value. They do not change the delta G, the difference in the free energy between the reactants and the products. Therefore, it is understood that they do not affect the K equilibrium or the equilibrium constant of the reaction. In other words, they do not affect the thermodynamic parameters of the reaction, but they allow for the faster attainment of equilibrium by increasing the rate of both the forward as well as the reverse reactions. So many a times it is not understood by students that the K equilibrium or the delta G of the reaction is completely unaltered by the catalyst by and large. And what the catalyst affects is the activation energy. Most biological reactions are extremely slow when uncatalyzed. For example, a simple glycolytic enzyme phosphate isomerase. The reaction, when it is carried out under uncatalyzed conditions, the T half, that is the time required for 50% of the reactants to get converted to product, is 1.9 days. This is pretty fast actually as compared to some other examples that are given here. For example, the enzyme carboxypeptidase or the reaction catalyzed by this enzyme. It's a simple breakdown of a peptide bond. It takes 7.3 years to get half completed. The enzyme nuclease, it takes several thousands of years for the reaction to get 50% completed, and certain other enzymatic reactions, for example, take millions of years to get even 50% completed. So this shows that many of the reactions which are of significance in life are extremely slow and the rates are incompatible with the living systems. For an E. coli to double every 15 minutes, it cannot afford to wait for a nucleotide to get synthesized in millions of years. So the important aspect of enzymes is their immense catalytic power. They are very extremely high reaction rates, or they can speed up the reaction rate much higher than the uncatalyzed reaction and make it compatible with the life and the necessity for the living systems. Besides that, enzymes are able to carry out these reactions under much milder conditions than chemical catalysts. They require lower temperatures, they require normal, near neutral pH, and so on. Another important feature about enzyme is their reaction or their specificity. And last but not the least is the importance as far as their regulation is concerned. They can be regulated as per the needs and the demands of the cell. This table shows some of the catalytic constants for some of the enzymes and is compared with the non-enzymatic reaction rate. So the non-enzymatic reaction rate given in terms of per second, that is the number of molecules converted from reactants to the products per second, is given in this column here, and this is the similar unit expressed when the enzyme or the catalyst, biological catalyst, is added into the system. The rate enhancement, which is the ratio of the two, shows us that the enzymes can enhance rate from 1,000,000 times to about 10 raised to 14 times, and the particular reaction that I gave you example of, OMP decarboxylase, for example, it can bring about rate enhancement of up to 10 raised to 17 times. So this is the high catalytic power of enzymes, which one has to remember that they are extremely good at catalysis. Models that explain specificity of the enzymes. The earliest model was proposed by Emil Fischer in 1910. And this model is famous as the lock and key model. The lock and key model states that the enzyme is like the lock and a substrate fits in exactly like how a key fits into the lock. And this indicates actually that there is a structural, that is geometric, and chemical complementarity between the enzyme and substrate so that they react in a perfectly complementary manner. The lock and key model was further expanded or modified by Koshland in 1958 to give what is known as the induced fit model. In the induced fit model, the enzyme, or the free enzyme rather, does not have a site which is exactly complementary to the substrate, but upon binding, the complementarity actually develops or increases, and the enzyme fits itself or according to the substrate. So this Koshland model and the Emil Fischer model are particularly noted for the difference in the flexibility of the enzyme. In the lock and key model, the enzyme is considered rigid like a lock, and in the Koshland model, it is considered flexible, more like a glove in which the hand fits. All enzymes have a particular site at which the reaction takes place. This site, known as the active site, includes the binding and catalytic residues. The characteristic features of active site are that they are actually a very small part of the total dimension of the enzyme. And this small part is developed because of the folding of the enzyme, and therefore it is a three-dimensional entity, and it can be dynamic. There can be changes because of the flexibility of the enzyme. Usually active sites are either clefts or crevices on otherwise a globular protein or spherical type of a molecule. The substrate are bound at this particular site, that is the active site, and they are bound by multiple weak non-covalent bonds. The active site also poses like a unique microenvironment where the properties may be different than the bulk environment, the bulk environment being aqueous in nature. The microenvironment may have properties which are not exactly the same as an aqueous environment. And this microenvironment is defined by the amino acids that line the active site. Because of the change in the environment, or because of a unique nature of the microenvironment, the R groups of certain amino acids placed in this microenvironment may differ than what they behave in aqueous solutions. For example, the pKa's of the R groups may be shifted when they are present in the active site, and this may be particularly important for the reaction to take place. Now, coming to some of the questions that are based on these aspects or these concepts. This model question asks, which of the following statement is true? Enzyme catalysis of a chemical reaction increases the forward and reverse reaction rates, decreases delta G naught so that the reaction can proceed spontaneously, increases the energy of the transition state, and decreases the entropy of the reaction. By scrutinizing these options, it is very clear that the enzyme does nothing to the delta G naught. It actually does not increase the energy of transition state, it decreases the energy, and it does not do any change in the entropy of the reaction. So therefore, the correct option is increases the forward and the reverse reaction rates. In another problem, model question, it is said that in an intact cell, the free energy change, it is delta G, associated with an enzyme catalyzed reaction is frequently different from the standard free energy change, delta G naught prime, of the same reaction, because in the intact cell, A, activation energy is different, reaction is always near equilibrium, enzyme may be regulated allosterically, and reactants are not at 1 molar concentrations. So again, going back to the question, it is very clear that the question is testing about the difference between delta G and delta G naught prime. So if we remember the definition of delta G naught prime is the free energy change of a reaction under standard conditions. These standard conditions are that all reactants and products are at an initial concentration of 1 molar, pressure of 1 atmosphere, and temperature is 25 degrees. So in an intact cell, it is quite possible that the standard conditions are not met. Particularly, the concentration of reactants being at 1 molar seems most unlikely. Therefore, going back to the options, we see here that activation energy is different. I think that is not the correct answer. Reaction is always near equilibrium. That also is not an correct option. And enzyme may be regulated allosterically. This will not cause a difference in the parameter that is mentioned. So the correct option seems only that the reactants are not at 1 molar concentrations. In another model question, it asks that for the enzyme catalyzed reaction S to P, K equilibrium equal to 5, which of the following curves best represents the change in substrate that occurs with time when substrate is added to a solution containing the appropriate enzyme? So looking at the graphs that are provided, the first thing that we should notice is what is plotted on the y and the x axis. So we see here that the substrate concentration with respect to time is plotted in all the options that are provided. So here in this question, the question that is asked is that when the substrate is mixed with the appropriate enzyme and the reaction has a K equilibrium of 5, how does the substrate concentration change with respect to time? So if you recall, what I had already mentioned is that upon addition of the appropriate enzyme, the reaction attains equilibrium faster. So in other words, this reaction is going to attain equilibrium at a certain time, and at equilibrium, once the reaction attains equilibrium, there is no net change in the substrate concentration. So we see here in all these plots that upon incubation, at a certain point, there is a plateau that is reached and there is no further change in the concentration of the substrate. So this states that the equilibrium has probably been reached at this time. Now all of these graphs, 3 of them are similar except for this one, the option D, which suggests that there is no change in the concentration of the substrate with time. So since the reaction, the K equilibrium of the reaction is 5, what we have to think about for the solution to this problem is that when the K equilibrium is 5, therefore, at equilibrium, the product concentration divided by substrate concentration is 5. Or in other words, during equilibrium, product will be 5 times that of the substrate concentration. So at equilibrium, product concentration is 5 times higher than the substrate concentration. Now we go back to the graphs and we see, for example, that the starting concentration of the substrate in all these plots is close to 1. So we assume that the starting concentration of the substrate is 1 millimolar. At equilibrium, therefore, the equilibrium concentration of substrate plus product is always going to remain what was initially the concentration of substrate to begin with, that is 1. So now we have these two equations, that S equilibrium plus P equilibrium equals 1 and P equilibrium equals 5 times S equilibrium. So we can solve these two equations in such a way that S equilibrium plus 5 times S equilibrium is equal to 1, or in other words, 6 S equilibrium equals 1, or S equilibrium equals 1 by 6 or 0.167. What does this value of 0.167 tell us? It means that when the system attains equilibrium, the substrate concentration at equilibrium is going to be 0.167, after which there will be no further change in the substrate concentration. So we go back to the graphs and we will see that, if I just go back, that in graph A, the substrate concentration almost becomes 0. In graph B, C, the substrate concentration is above 0.5. This, of course, we have already ruled out. And in this graph B, the substrate concentration is close to what we expect from our solution of this problem, that is between 0.5 and 0, that is 0.167. So the graph A shows S equilibrium to be 0, graph C shows S equilibrium above 0.5, and graph C shows S equilibrium is not changed, and it is the same as ST. And only graph B approximates to the correct value of S equilibrium, and therefore we can say that the correct answer is graph B. Now we come to another aspect about enzymes, and that is enzyme classification. Enzymes are classified by the nature of the reaction that they catalyze. And all biological reactions can be actually grouped into 6 different categories. And these categories have been given an EC number. This EC number is in a particular order because that number is going to determine which particular class the enzyme belongs to. So the Enzyme Commission gives a unique 4-digit number to each enzyme, of which the first digit indicates the broad class to which the enzyme belongs. So all oxidoreductase catalyzing oxidation or reduction reactions belong to class 1. EC2 is transferases, that is group transfer reactions. EC3 is hydrolases that catalyze the hydrolytic reactions. EC4 is lyases, EC5 is isomerases, and EC6 is ligases. And I would advise students to remember these broad classes of enzymes in the particular order, because this order gives you the first digit of the group to which the enzyme belongs. Oxidoreductases can be broadly again classified into dehydrogenases, oxygenases, monooxygenases, dioxygenases, hydroxylases, peroxidases. So we see here that there are different ways in which oxidoreduction type of reaction can be carried out, directly with oxygen as a substrate, or with the transfer of an electron, or introducing a hydroxy group, and so on. So the second digit of the EC classification system is based on these further differences between the oxidoreductases. Transferases are enzymes which transfer a group from substrate A to substrate B. They could transfer a phosphate group, or a sulfate group, or a keto and aldehyde group, amino group, monosaccharide group, nucleotidyl group. So DNA polymerase, for example, is a transferase. Hydrolases are enzymes which cleave bonds by the addition of water molecule to the bond that is broken. So many types of enzymes are hydrolases, glycosidases, phospholipases, peptidases or proteases, nucleases, phosphatases. They are classified on the basis of the bond that they hydrolyze. An important group of enzymes, which is sometimes a bit tricky to identify, are the lyases. Lyases also carry out cleavage reaction. They can break CC bond, CO bond, CN bond. But unlike the hydrolases, they do not do so by the addition of a water molecule, neither by oxidation or reduction. In most cases, the product that is generated is an unsaturated molecule plus an XH species. Or in other words, in a very simple way, we can say lyases remove a group from the substrate, but they leave a double bond behind. So to understand a lyase reaction, it's very important to know the structure of the reactants and the products. So carbon-carbon CC lyases, for example, decarboxylases, remove carbon dioxide or CO2. Carbon-oxygen lyases or dehydratase dehydratases remove water molecule. So in a reverse direction, the removal of water could rather appear like a hydrolytic reaction, but there are ways to identify a lyase reaction. The carbon-nitrogen lyases, for example, ammonia lyases, remove ammonium. Isomerases, this group of enzyme basically carry out reactions which are intramolecular in nature. So they can carry out intramolecular electron transfers, group transfers, and changes in the molecular structure. So the product and the reactant have the same molecular formula, but they differ in the bond connectivity or the spatial arrangements. So these isomerases can shift or change molecules between positional, geometric, or optical isomers. The last group of enzyme ligases, they form a bond between reactants, mostly using ATP as a co-substrate to drive the reaction. So they basically can catalyze the joining of two large molecules by forming a new chemical bond, usually with the accompanying hydrolysis of a small pendant chemical group on one of the larger molecules. A stereotype reaction is written here where AB plus C gives rise to AC plus B. An aspect of enzyme classification is also enzyme nomenclature. The reaction that is shown here is the reaction for lactate dehydrogenase, which is the common name for this enzyme, but a systematic name for this enzyme would include the names such as L-lactate NAD oxidoreductase. So we see here the substrate, the cofactor, and the fact that it is an oxidoreductase. So there are different rules for writing the names of different classes or different major groups of enzymes, and some of these can be remembered by the students because many a times questions are asked regarding this. Coming to some questions based on the enzyme classification, we see here that there is a match the following type of a question where the different types of reactions are given, the reaction is given here and the group to which the enzyme belongs is given here. So we have kinases, lyases, synthases, phosphatases, and it should be very clear to you now that what are the kind of reactions that these groups of enzymes catalyze. So we see kinases have transfer phosphate group to biomolecules, lyases cleave bonds by elimination, synthases make large molecules from small molecule, and phosphatases remove the phosphate group from biomolecules. Coming to another question, the systematic name for the enzyme catalyzing the following reaction is fumarate lyase, fumarate hydrolase, malate hydrolase, malate hydrolase, and malate hydrolase. So taking a look at this reaction, we see here that the reaction involves conversion of fumarate to malate with the involvement of a water molecule. And the options that are given here show that we have to choose between whether this enzyme is a hydrolase or a lyase. So as I had told you that the key difference between a hydrolase and a lyase is that a group is removed by leaving a double bond in the original molecule. So if we look closely at this reaction, we see here that a group is removed from malate. That group is a water molecule, and what results in this residual molecule is a double bond between the CC to give rise to fumarate. So by understanding this aspect of this reaction, we see that the water molecule, although at this stage, if we look from the forward direction, is added, but there is no bond that is broken. So it cannot be a hydrolase, and it has a good chance of being a lyase. So the lyase nomenclature is the molecule or the substrate and the group that is removed and lyase. So we have the best option here, or the correct option I would say, is malate hydrolyase. So the nomenclature to be followed is substrate, the group removed, followed by lyase. So if we correctly look at this reaction in this direction, it could be understood that it is malate hydrolyase. Another question regarding enzyme classification is, to which group of enzymes does the enzyme aconitase catalyzing the following reaction belong? A. Hydrolase, C. Lyase, B. Isomerase, C. Lyase, and D. Transferase. So we have to take a good look at the structure of the reactant and the product, or the substrate and the product, and we see that citrate and isocitrate are isomers. There is only a small shift here between the hydroxyl and the hydrogen groups. Otherwise, the molecular structure of isocitrate and citrate, the formula remains the same. So in that way, there is a possibility that this is an isomerase, and definitely it is not a hydrolase or a transferase. But if you look at the enzyme aconitase, the name aconitase, the name does not really befit this conversion of citrate to isocitrate. So there is something more to it. And in fact, aconitase is a lyase. So the question arises, why is aconitase a lyase and not an isomerase? Because citrate and isocitrate are isomers, and the enzyme aconitase interconverts the two, yet it is considered a lyase. So this question, the answer to this question lies in the fact that this enzyme catalyzes the reaction conversion of citrate to isocitrate through an intermediate which is cis-aconitate, whose structure is given here. So using this hint and knowing the details about the actual reaction that is catalyzed by aconitase, we see that aconitase actually converts cis-aconitate to either citrate or to isocitrate with the involvement of a water molecule. So the reaction is very similar to that of the fumarase enzyme, and we see here that the water molecule is removed from citrate to give rise to cis-aconitate, where there is a double bond generated, or it could be removed from isocitrate to give rise to cis-aconitate. So the enzyme binds to citrate, converts it to cis-aconitate. The cis-aconitate is released from the enzyme active site, and it could again bind to the enzyme active site and get converted to the isocitrate. So the enzyme aconitase catalyzes the conversion of either citrate or isocitrate to aconitate, and the substrate is released after each step. And that makes it aconitate hydratase, and the reaction or the enzyme belongs to class 4, EC4, that is a lyase. So this was a little tricky kind of a question, because from the look at the substrate and the product, it could not be made out that this is a lyase until we understand in detail the reaction mechanism that aconitase follows. Now I come to the last aspect of part 1, and that is enzyme assay or enzyme activity measurements. In order to understand activity measurements, the first thing that we need to understand are reaction orders with respect to the concentration of reactants. So the law of mass action states that the rate of a reaction is always proportional to the concentration of the reactants. Now here are some different types of reaction orders based on how they are dependent on the reactant concentration. So a zero order reaction has a rate equation where the rate is equal to K. K is a rate constant, and the reaction we see is independent of the parameter, that is the reactant or any reactant molecule. So it is independent of the concentration of any of the reactants. So that is a zero order reaction where the reaction becomes completely independent. Even if you add large amount of reactants, it does not affect the rate. A first order reaction, on the other hand, has rate equal to K into A, where the reaction rate we see increases linearly in proportion to the concentration of A. In a second order reaction, the rate is dependent on the concentration of two reactants. They could be two different reactants, or they could be two molecules of the same reactant. So this is also a second order rate reaction. This is also a second order rate reaction, but here the rate is dependent on the concentration of the two reactants, each one of them linearly. If any one of them is in excess, if for example B is in large excess, it will become a pseudo first order reaction where it will depend only on the concentration of A, since B is in large excess. So that becomes a pseudo first order reaction, although it is a second order reaction in reality. This reaction where two molecules of A are combined to give rise to the product in one cycle of the reaction, hence the rate is proportional to the concentration of A, but not linearly. It is proportional to the exponent or factor raised to 2 of the reactant concentration. The important inference from this type of understanding is that if the reaction rate is linearly proportional to the concentration of A, the reaction rate can be used to estimate the concentration of A. So if we have a rate of X, we can determine what is the concentration of A from either knowing the K value or by plotting a graph whereby we can measure the concentration of estimate the concentration of A. In the second order reaction of this kind also, A can be estimated if B is in large excess, and vice versa, B can be estimated if A is in large excess. Now how does this relate to the enzyme assays? So if we say that the reaction rate is proportional to not only the substrate concentration but also enzyme concentration. So in an enzyme assay, we want to measure the enzyme units or the enzyme activity, and in that case, therefore, we must keep substrate in excess. So when the substrate is in excess and we plot a graph of enzyme concentration versus rate of the reaction or the product formed, it will show a linear first order type of a reaction. So this forms the basis for determining the enzyme concentration. So if we determine the rate of the reaction, we can get to know what is the enzyme concentration. With high substrate concentration, the reaction becomes zero order with respect to substrate and proportional in the first order to the enzyme concentration. So we see here that this is a slightly different graph than this. Here the rate of the reaction is plotted. Here the product concentration is plotted with respect to time. And we see again a linear dependence or linear generation of the product with a particular concentration of enzyme, and with double the enzyme there will be double the product formation at the same time. At a given time, with 1x enzyme there will be certain amount of product, with 2x there will be twice that much amount. So this is also a graph which shows that the reaction or the product for concentration can be a measure of the enzyme concentration if the substrate is in high quantity. So this forms the basis of enzyme quantification in terms of units. Usually the units of the enzyme are defined as micromoles of product formed per unit time. In reality, when we carry out the the assays, it is possible sometimes that substrate becomes limiting, and this happens particularly with longer incubation times. So if we take a look at this graph a little closely, this is the product concentration versus time, and ideally this is the dashed line, ideally how it should go. But in reality, most of the time the graph tapers down, and the reason for that is that substrate is limiting. Although we started with high amount of substrate, it cannot remain a high amount for extensive incubation periods, and after a certain point the substrate becomes limiting. So what this plot tells us is that the time A to B, the time zone from A to B, is ideal for assaying the enzyme, but not from B to C, because here if we use this product concentration, we are underestimating the enzyme because we have gone out of the zero order reaction. So this happens because the substrate becomes limiting. Another aspect about enzyme assays is to distinguish between enzyme activation and enzyme induction. Activation is brought about by direct interaction of the molecule with the enzyme protein, and induction is brought about by change in the gene expression by that particular molecule, which we call as the inducer in this case or the activator in this case. A notable difference between the two is that in activation the enzyme concentration remains unaltered, but its activity or its efficiency is increased. Here the enzyme concentration is increased, and as a result its activity is increased. In both cases activity increases, but here because the enzyme protein is made in more quantity, and here because the enzyme has been converted into a catalytically more efficient form. Activation can be brought about by both non-covalent interactions or covalent modifications such as phosphorylation, adenylation, and certain other modifications which are known. And induction, we know, can be brought about by either de-repression or transcriptional activation of the gene which encodes the particular enzyme. In exactly the same manner, we can distinguish between inhibition and repression of the enzyme. Now coming to a few problems based on these concepts, we have here a question which states conversion of substrate S present in large excess to product P by an enzyme increased in a linear fashion for about 10 minutes, after which it was seen to plateau. This is because the enzyme has reached its Km, reached its Vmax, has been inactivated after 10 minutes of activity, and switched its substrate specificity. So again, if we look at the question little more closely, the substrate S is present in large excess, and the enzyme converts it to product P in a linear fashion for about 10 minutes. Now, Here, the option that it has reached its Km is very unlikely because a Km cannot be actually reached with time. Km is reached when a particular substrate concentration is added into the reaction mixture. The reaction may proceed at when that is how you actually estimate Km. So Km is a constant and it is not reached with time, I would say rather. The enzyme has reached its Vmax. This also we can see, let us first rule out some other unlikely points. So the enzyme has switched its substrate specificity. This again never happens unless some modification of the enzyme is involved, and that is unlikely because this is in an in vitro condition. The enzyme reached its Vmax. This also cannot be correct as the substrate is already in large excess. So the enzyme is operating at Vmax from the beginning. The best answer to me is answer C, that it might be inactivated after 10 minutes under the incubation conditions in which it was held. And this is quite likely. Many a times while doing the assay, the enzyme slowly gets denatured because the optimum condition for the enzyme to remain active need not be the optimum condition for it to show the best activity. So the correct answer, in fact, in some of the answer books is given as B. To me, this is not the correct answer, and C is the best answer. This is another question based on enzyme activity. Here, the question states, in order to study the enzyme activity of the enzyme X, cells were treated with glucagon, dibutyryl cyclic AMP, and glucagon plus H8 at a dose required to achieve maximal effect. The activity and concentration of enzyme X were then assayed in dilute cell-free extracts. Dibutyryl cyclic AMP is an analog of cyclic AMP that diffuses across the cell membrane more easily than does cyclic AMP itself. H8 is a selective inhibitor of protein kinase A. The results are shown in the following table. So if you look at this table, the table shows what kind of treatment was done to the cells and what were the enzyme activity in terms of units and what is the enzyme concentration in terms of microgram per ml. So enzyme concentration is obviously measured by some other means other than using the enzyme activity units, but this concentration is in terms of micrograms of enzyme protein per ml of the extract. So we see here that when the agent is added, no agent is added, the enzyme activity is 10. When glucagon is added, the enzyme activity increases to 100 units. Independently, dibutyryl cyclic AMP also can increase the enzyme activity to 100 units. But when H8 is added along with glucagon, it prevents glucagon from increasing the activity to 100. When we look at this column here, the protein concentration ranges from 9 to 12 micrograms per ml. So let us go to the question that is asked regarding this data. So this is actually nothing but how the experiment was done and what were the results that were obtained from this particular experiment. Now on the basis of these results, there will be some questions which are usually posed which ask us to infer about this data. So some of the inferences that we have already made are these. So we note that the cells were treated with the various agents, not the cell-free extract, and therefore the various agents could act at different levels That means at gene expression or at protein expression levels. The second aspect that we have noted already is that the enzyme concentration in terms of micrograms per ml remains more or less same under all the treatments. So we infer here, therefore, that there is no induction or repression, that is, no change in the gene expression or transcription brought about by any of the agents. We also infer that the enzyme turnover in terms of its degradation does not happen. So we assume that this range from 9 to 12 is within the range of experimental error, and that basically enzyme concentration remains virtually unchanged, but there is a drastic change in enzyme activity units. So on the basis of that, we conclude that there is no induction or repression, and neither is there any degradation of the enzyme that happens. So now the question that has been asked regarding this data is, it can be inferred from these results that glucagon most likely acts to cause translocation of the enzyme X to the lysosome, increase the catalytic efficiency of the enzyme X, inhibit degradation of the enzyme X, stimulate transcription of the gene for enzyme X. So this question has to do with the role of glucagon in the particular experimental setup. So if we go back again to this table, we see that glucagon brings about 10-fold increase in the enzyme activity and hardly any significant change in enzyme concentration. So that is what we have to observe here as compared to the control, or when no agent was added. So that is the observation that glucagon treatment increases enzyme activity by 10-fold, but it does not change the enzyme concentration significantly. So what are the options that we have here? That glucagon causes translocation of the enzyme X to the lysosome. So this option A, there is absolutely no reason to believe that there is something to do with lysosome, because there is no reference or connection to lysosome is mentioned. Another option which we can eliminate in this elimination order is that it stimulates transcription of the gene for the enzyme X. We already inferred that the data suggests there was no induction or repression, no change in transcription. Option C is inhibits degradation of the enzyme X. So again, data shows that there is no degradation seen in terms of the protein amount. And option B, therefore, is the correct answer, that it increases the catalytic efficiency of the enzyme X. For this same data, another question that has been asked is, it can be inferred from these results that the changes in the enzyme X, activity of the enzyme X, are mediated by allosteric interaction of intermediary protein with H8, proteolytic cleavage of an intermediary protein, a phosphorylation probably catalyzed by protein kinase A, a cyclic AMP independent signaling pathway. So we infer from this that this question has to do with the role of H8, cyclic AMP, and protein kinase A. So we have to go back to the question a little more and scrutinize it a bit more carefully. We see that dibutyryl cyclic AMP is an analog of cyclic AMP that diffuses across the cell membrane more easily than the cell cyclic AMP itself. And H8 is a selective inhibitor of protein kinase A. And if we see what these two agents do to the enzyme, we find out that dibutyryl cyclic AMP stimulates the enzyme activity, whereas H8 inhibits it. So we infer that dibutyryl cyclic AMP increases enzyme activity as much as glucagon does. Inhibitor H8 prevents the activation. H8 is known to inhibit protein kinase A, therefore protein kinase A is positively involved in the activation of enzyme X, and protein kinase A may activate enzyme X directly or indirectly by phosphorylating another protein. So if we eliminate the options in terms of their unlikeliness, we eliminate option D, option B, option A, and we have only option C as the correct answer, that a phosphorylation event probably catalyzed by protein kinase A is what affects the enzyme activity. So thank you, friends. This brings us to the end of part 1, and I'll see you later in part 2.
Vande Gujarat.